now we'll listen to Østergaard Klein. Caroline is a PhD student at the Research Group for Food Microbiology and Hygiene. And you'll be talking about sustainable methods to stabilize and utilize cultivated kelp in high quality and safe food products. And then from now on, Tim will be presenting the next speakers after this one.
Hi, I'm Caroline, and today I will present to you my PhD project called Sustainable Methods to Stabilize and Utilize Cultivated Kelp in high quality and safe food products. So I will just give to you a short background, and then I will share the results so far of my first and large experiment that I'm going on. Okay? Yes. So as for many of you in this room, we don't include seaweed in our diet. But in order to do so, we need new products to be developed. And these products need to be attracted to consumers with regard to taste, nutrient content, shelf life, and food safety. And this requires the best quality of kelp. And here a classic bottleneck problem happens because you see the harvest occurs over a few weeks in the late spring and early summer in the northern countries, which is a short period for a harvest. And then the kelp needs to be stabilized within 7 days in order to avoid food spoilage, food lost, and food waste. Today, the common methods to stabilize kelp is by drying or freezing at specialized facilities. But what about the low technology methods like preserving with salt and or acid? And this is where I fit in the picture. So my hypothesis is that low energy intensive preservation methods can be formulated and optimized to modify the key characteristics of kelp, so meaning the pH and the water activity, to inhibit or inactivate microorganisms and enzymes to extend the shelf life of high quality intermediary products. And by intermediary products, I mean the stabilized kelp, which will then later be used by food producing companies in the final food product. So, to test my hypothesis, I have performed a storage trial with freshly harvested sugar kelp, which I have cut and I have salted it, and then stored at 3 degrees. So for 3 months, and still with a few samplings to come, I have looked at the microbial and physiochemical changes. So, when you mix the cut kelp together with salt, you reduce the water activity. And so, the higher the concentration of salt, the lower the water activity gets. And water activity is important for the microbial growth. Depending on the microorganisms, they can grow to a certain extent. So most bacteria, they cannot grow below a water activity at 0.90, while other microorganisms as yeast and mold, they can, also together with some bacteria as Staphylococcus aureus and halophilic bacteria, which by its name means salt-loving bacteria. When describing the bacterial growth over time, the water activity takes part in this inactivation or inhibition of the bacterial growth. So adding 10% salt or more to the kelp, you see a reduction in the number of cells compared to the starting point. And with these level of salt, they correspond to water activity around these 0.90 and below, where I just told you most bacteria don't grow. I have had great participation from people from the research group because I wanted to get an idea about the presence of off odor during this storage trial of these salt-treated silver kelp samples. And I asked them to rank the smell. So 1 meant no off odor, 2 was weak, and then 3 was a strong off odor. And This one on the left is from one of the first days compared to the latest sampling. And as you can see, a high concentration of salt is needed to keep this neutral smell. I also performed a color analysis where, again, at each sampling during this storage trial, I then saw that the untreated sugar kelp, so it doesn't have any salt, it was significantly different from those treated with salt, while there was no significant differences between those treated with salt. So regardless of the salt level, they kept a green color. I'm in the preliminary phase of making the next big experiment, so another storage trial. And here I will apply different combination of salt, which I've learned from the first experiment. And then I will add acid, based on literature, to cut frozen sugar kelp and wing kelp from Greenland. Thank you for your attention. I have had great support from my supervisors and lab technicians, so I'm looking forward to continuing working with them. Thank you.
Wait a second. Thank you very much. Are there any questions? We have some time for questions if you would like to ask something. Yes?
Where can I buy kelp in Denmark, in stores?
I mean, from my knowledge, most of the kelp is dried or freezed, but I try to preserve it as fresh. And at the moment, I don't think you can buy freshly harvested kelp unless you buy it from those who harvest. So, for example, at Østergaard, there are some companies, Danish companies, that do have it.